నమస్కారం. డాక్టర్ బిఆర్ అంబేద్కర్ సార్వత్రిక విశ్వవిద్యాలయ విద్యార్థులకు స్వాగతం.
Dear students, today we are going to learn about a topic from course 8 MSc second year, that is genetic engineering and its prospects. You might have already learnt about fruits which have different flavor and color, like an apple which looks like a banana, and tomato which is purple like brinjal, and fluorescent fish which glows in night, and fluorescent pigs and other kinds of cattle. How do you think it is possible? This is because of what is called as a modern branch called genetic engineering. Now let's learn about what is genetic engineering. Now it's a modern branch of biotechnology. Here we define genetic engineering as a technique which involves breakage of DNA from a desired organism and isolating and inserting the same into another organism where it expresses the same character. This particular term genetic engineering was first coined by Jack Williamson in 1951. Now it is a logical progression from the conservative method of hereditary, that is previously the breeders used to use the technique of artificial breeding to bring out the required traits in whether it may be plants or whether it may be animals. Now this is completely in highly advanced science. The product thus obtained by this one is called as a recombinant DNA. Now these transformed products are called as either transgenics or transplasmids, as transgenic means transformation in a part of the nucleus and transplasmatic means transformation in the plastids. Now after this transformation, the selected cells are isolated and cloned to produce multiple copies of the desired traits. To produce these copies, it is possible because of presence of vectors. Now let's learn what is a vector. A vector is an agent that can carry a DNA fragment into a host cell. Now when we talk about vectors can be various types. Primarily it can be a plasmid, it can be a virus, or it can be any other agent which has capability to carry the desired genes from one organism to the target organism. Now let's, based upon the function, we come across two kinds of vectors, that is the cloning vectors as well as the expression vectors. Now what is a cloning vector? A cloning vector is one which is used to produce multiple copies, the two identical copies of the recombinant DNA. Whereas an expression vector is one which is used for expressing certain gene in the DNA fragment. Below you can see the two pictures of the same. Now when we come to vectors, as I told you early, the commonly used vectors are either plasmids or the phages. Now what is a plasmid? Plasmid is an extra chromosomal DNA which is present in the cytoplasm of the cell. Usually this is found in the bacteria. Now let's learn few characters of these plasmids. They are circular, they are double-stranded DNA which are found in bacteria as well as a few eukaryotic organisms. The size of the plasmid is few kb to 100 kb in length, and the plasmid encoded traits include resistant to antibiotics, degradation of complex organic molecules. It has a capability of production of bacteriocins, enterocins, antibiotics, DNA restrictions and modification enzymes, etc. Now examples of this plasmid are PBR322, UC1819, B blue script, etc. You can see the images of the plasmids here. You can see the circular plasmid which is present in the cytoplasm of the bacteria. Now let's learn about what are phage vectors. Now a bacteriophage is actually a virus which attacks only bacteria, that is bacteria eating virus is called as an bacteriophage. Now this is possible because that virus can lyse, that is it can dissolve the cell wall of the bacteria and enter into it. Now through the two stages, that is either lytic cycle or lysogenic cycle, the phage chromosomes incorporates itself into the bacterial DNA, which is now called as a prophage. Example of the phage vectors are E. coli, lambda phage, phage M13, etc. Now compared to the plasmid, phage vectors are more advantageous since they can be directly tested on the appropriate bacterial lawn. They are more efficient because they can clone larger DNA molecules, and they are very easy to screen once the recombination takes place. You can see the images of the phages here, the E. coli phage, lambda phage and M13. Besides the plasmids and the phages, the other kind of vectors which are usually used in genetic engineering include cosmid vectors. A cosmid vector is a one which is an hybrid between a plasmid and a phage virus, whereas phagemids are once again hybrid viruses which contain a part of M13 phage in them. Besides that, we also come across eukaryotic plasmids like shuttle vectors, yeast vectors, yeast plasmids, bacterial yeast chromosome, artificial chromosome vectors, etc. Now, how does the mechanism of genetic engineering occurs? I have here, you can see three examples here. The first example is insertion of ringworm virus into a papaya plant. Now, ringworm causes disease in papaya, but the resistant gene is present within the ringworm virus itself. So, the gene which expresses such kind of disease resistance is isolated from the ring virus, and through the help of a vector, it is inserted into a papaya cell. After its insertion, the papaya cells are cloned to produce numerous copies of it and later regenerated into a disease resistant papaya plant. Now let us see another example of the genetic engineering. Here you can see a tobacco plant which is luminated. It is a recombinant plant, and this recombination is because of the one gene which is present in the fruit fly, which is a gene to express luminescence in the fruit fly. This gene is isolated from the fruit fly, and through the help of an vector, it is inserted into the tobacco genome. After the transformation, the transformed cells are isolated, multiplied and regenerated to produce an illuminating tobacco plant. The third example and the most important one is insulin producing bacteria. We know that India stands number one in diabetic patients. Now, because diabetes is the resultant of a lack of insulin production, here we can see that insulin is produced in the alpha cells of islets of Langerhans. So, the gene which codes for insulin is isolated from the alpha cells and it is inserted into a bacterial genome. After the transformation, the bacteria are cloned and multiplied. Now, this bacteria, since we know that bacteria rapidly divides, it can produce insulin on a large scale, which would help the diabetics to control the disease. Now, just we will once again recollect the steps involved in recombination here, which helps in process of genetic engineering. There are 8 steps here. The first step is isolation of the required gene or the desired gene from the required plant. We have to first identify the desired gene which has to be isolated, and after isolation it is inserted into a transferring vehicle, that is a vector which can be either a plasmid or it can be a phage or any other suitable vehicle. The phage or the plasmid will carry this required gene into the target cell, where the gene is inserted into the target cell genome, and here it is cloned and multiplied, and after multiplication these are isolated and regenerated into whole plants under controlled conditions. After they are successfully exhibit the trait, these are released into the market. the last and the most important one of this concept is the safety assessment. Genetically engineered products are always been a controversy, hence safety assessment from the government, approval from the government is essential for their usage and release into the market. Now, after the desired gene is multiplied, there are 3 steps for it, that is selection, screening and recombinant multiplication. Now, there are various methods to identify the transformed cells. Basically, there are 3 procedures. First one being, there are specific enzymes or something called as selectable marker genes or reporter genes, which can help in identifying the desired gene is whether has been incorporated into the target or not. And besides that, there are certain analytical procedures like PCR technique, bioassays, immunological techniques, spectroscopy, etc. The third one and the latest being DNA chip technology. Through these procedures, we can easily identify whether the required gene has been incorporated into the target or not. Thus, identified transformed cells are allowed to regenerate into a complete organism by using micropropagation cloning techniques. Here you can see the pictures of marker genes, reporter genes, DNA chip and PCR techniques which are used in identification. Now, after this procedure, let's try to understand what are the prospects of genetic engineering. Here, the applications of genetic engineering are wide in number. Here, the first example of genetic engineering, the first one is Bacillus thuringiensis was the first genetically engineered product. And if we talk about animals, the mouse was the first animal which was first genetically engineered. The applications of genetic engineering are widely seen in agriculture, medicine, dairy industries and other allied areas. Now, once again we can recollect what is a genetically transformed organism. A genetically modified organism or a genetically engineered organism or living modified organism is one whose genetic material has been altered using genetic engineering techniques. Now, it aids in creating crops with desired traits like high productivity, high quality, incorporation of desired characters, resistance to pests, insects and viruses, tolerance to herbicides and stress resistance, etc. Few examples of transgenic plants, animals we would see now. Now, transgenic is a science of intentionally introducing a foreign gene or genetic element into the genome of a target organism, which creates transgenic animals, plants, bacteria and viruses. Now, first we will see genetically engineered crops. These are famously called as GM crops. The popular ones what we can see now in a market are the golden rice, golden mustard, cassava, multivitamin corn, GM rapeseed, protein rich potato, GM potato, maize, BT cotton, etc. Among them, the golden rice is one which is now fortified with vitamin A so that they can fight vitamin A deficiency. Now, cassava is an African tuber which is enriched with proteins, and in the same case multivitamin corn, here the genes are incorporated to produce numerous protein, more protein into it and more fiber into the corn. Now, let us see the next one. Now, biopharmaceutical is a recent concept. Bio means living, pharmaceuticals are medicinal elements. Now, here biopharmaceutical, under this there are 2 categories. Here edible vaccines and plant antibodies. Now, what are vaccines? Vaccines are any organism which is once administered into an individual will confer immunity against a disease. Now, edible vaccines are one what we take regularly as our food. So, edible vaccines are produced in genetically modified crops which holds a promise to produce cheap and effective protection against infectious diseases in the developing world. You can see the examples here: potato, bananas, apples can be effectively used as biopharmaceuticals or edible vaccines. Now, plant antibodies. They are bioactive molecules which provide immunity by producing functional antibodies. These recombinant antibodies include fully assembled immunoglobulins, antigen binding fragments, etc. The examples of plant antibodies are GM cereals, legumes, oil seeds, tubers, etc. Now, what is the source of biopharmaceuticals? Transgenic plants, cultured mammalian cells, bacteria, fungi all source of biopharmaceuticals which can be directly taken by the individuals so that they can have immunity towards various common to serious diseases. Now, the examples of the biopharmaceuticals that are present in these target individuals are erythropoietin, insulin, encophalins, alpha interferon, human serum albumin, etc. Now, let us see the transgenic animals. Here you can see various transgenic animals here. The glowfish is the first transgenic fish which is distributed as the pet, which would glow in the night. Here you can see fluorescent mice also which would glow in the dark, and you can see the salmon fish which is twice the size of a normal fish, and here you can see GM pig, goat, cows, etc. Advantages of transgenic animals. Why do we need them? They are the target to research against human diseases. Examples being various diseases like even AIDS disease can be treated by using the transgenic animals. One example I would like to quote here is that now that pigs has been genetically modified to produce human organs for transplantation, to produce industrial or consumer products, to enrich or enhance the animals' interaction with humans, to produce products intended for human therapeutics like production of different kinds of medicines, to improve the animal health such as providing disease resistance to them. Whether this modern science is a boon or bane. Now, the concept of genetic engineering is a recent one. Though it has a large amount of promise, we have to equally carefully weigh the pros and cons of it, because there has always been a constant debate on safety of using the GM products, whether they can be released into common market, whether any individual can consume it or not. So, there has been an long impact of GM food being deliberated by consumers, biotechnology companies, government regulators, NGOs and scientists. Though there is a scientific consensus that they are safe to consume, but still we have to think about the health hazard and the environmental risk involved in it. Though many countries are not able to accept the concept of GM food, compared to other countries, USA is more liberal about the GM foods. Institutes like GM Watch, Institute of Sciences and Society, Friends of Earth still strongly oppose the usage of GM foods. So, we have to measure both the pros and cons about it. Whether it is boon or bane, it depends upon the right usage and right target. Students, let us sum up the important points of genetic engineering. Now, we said that genetic engineering is a modern branch of biotechnology. It's a recent allied group of science. Now, it holds a lot of promise, as I said, because genetic engineering is a branch where it involves a technique of isolating a desired gene from a particular organism and inserting into a target organism. So, here we see a transformed or a recombinant organism with the modified gene. Now, how is it possible? How is the transfer possible from one organism to another organism? This is possible because of what is known as vector. Now, once the vector transfers the target gene into it, the gene is incorporated into target genome. After this insertion, the isolated gene produces numerous cloned copies of itself. Now such are called as clones. Now beside the plasmids and the phages, the other vectors that are used commonly in genetic engineering are the cosmid vectors, phagemids, shuttle vectors, yeast vectors, yeast plasmids, artificial chromosome vectors, etc. Now coming to the mechanism, that is, first we will identify the target gene of interest. Once it is isolated through a vehicle or a vector, which can be either a plasmid or a phage, it is inserted into the target organism. Once it is inserted, the DNA fragment enters into the gene of the target organism, and now it will start producing multiple identical copies called as clones in the genome. After the production of the clones, the desired clones are isolated and transferred into the tissue culture medium, and here multiple copies of this are produced. After it, regeneration of the same cell into a whole organism is possible in the tissue culture lab. Once it is attained, under the field area, control field area, these are allowed to grow into individual plants, and after screening for 2 to 3 generations, if they are found to be successful, they are released into the market. Now the one important thing that we have to remember here is about the safety assessment. When we are using a GM product, we should be assured that if it is consumed by the individuals, it should not produce any kind of ill effects, whether it should not be health hazard nor it should be a environmental hazard. Now we have seen various methods of identifying them, like using specific enzymes, reporter enzymes, analytical procedures like PCR, bioassays, etc. Now we will see what are its applications. Now genetic engineering applications are very wide because they are applied in almost all major fields like agriculture, medicine, dairy and allied sciences. Now we will see few examples of transgenic crops here. Here you can see the pictures of golden rice, which is enriched with vitamin A, multivitamin corn and grey mustard, which is rich in fiber, cassava, which is rich in proteins, colored potatoes, GM modified tomatoes, rapeseed oils with high oil content. Besides that, they can also act as biopharmaceutical, that is edible vaccines and plant antibodies. Now coming to transgenic animals, here the transgenic animals are very useful to us because they provide us with their meat is important, their products like wool is important, they are a source of milk, they also are a source of medicines to us. So transgenic animals are also important in it. Now whether it is boon or bane is in our hands, the technology should be handled carefully because it includes safety of the individuals and also pose a threat to the environment.
మా కార్యక్రమాలపై మీ సూచనలు సలహాలు తెలియచేయండి. మీ సూచనలు సలహాలు తెలియచేయవలసిన చిరునామా: డైరెక్టర్, ఆడియో విజువల్ ప్రొడక్షన్ అండ్ రీసెర్చ్ సెంటర్, డాక్టర్ బిఆర్ అంబేద్కర్ ఓపెన్ యూనివర్సిటీ, ప్రొఫెసర్ జి రామరెడ్డి మార్గ్, రోడ్ నంబర్ 46, జూబ్లీ హిల్స్, హైదరాబాద్ 500033.